restriction in the nucleus are used in genetic engineering to form recombinant molecules of DNA which are composed of DNA from different sources genomes. When cut by the same restriction enzyme, the resulted DNA fragments have the same kind of sticky ends, and these can be joined together end to end using DNA ligase. Normally, unless one cuts the vector and the source DNA with the same restriction enzyme, the recombinant vector of the molecule cannot be separated. Cloning vectors. In order to link the aligned DNA, vector needs to have very few, preferably single recognition site for the commonly used restriction enzyme. Presence of more than one recognition site with the vector will generate several fragments which will complicate the gene cloning. The ligation of alien DNA is carried out at the restriction site present in one of the two antibiotic resistant genes. For example, you can ligate a foreign DNA at the BAMH1 site of tetracycline resistant gene in the vector PBR322. The recombinant plasmids will lose tetracycline resistant due to insertion of foreign DNA but can still be selected out from the non-recombinant one by plating the transformants on tetracycline containing medium. The transformant growing on ampicillin containing medium are then transferred on a medium containing tetracycline. The recombinant will grow on ampicillin containing medium but not on the containing tetracycline, but non-recombinant will grow on the medium containing both the antibiotic. In this case, one recombinant resistant gene help in selecting the transformants, whereas the other antibiotic resistance gene get inactivated due to insertion of aligned DNA and helps in selection of recombinants. Selection of recombinant due to inactivation of antibiotic is a cumbersome procedure because it requires simultaneous plating on two plates having different antibiotics. Therefore, alternative selector markers have been developed which differentiate recombinant from non-recombinant on the basis of their ability to produce color in presence of chromogenic substrate. In this, a recombinant DNA is inserted within the coding sequence of the enzyme beta-galactose. This results in inactivation of the gene for synthesis of this enzyme, which is referred to as insertional inactivation. The presence of chromosomic substance gives blue color colonies if plasmid in the bacteria does not have an insert. Presence of insert results into insertional inactivation of the galactosidase, and the colonies do not produce any color. These are identified as recombinant colonies. Lesson of transferring genes into plants and animals from bacteria and viruses have known this for ages. How to deliver genes to transport eukaryotic cell and force them to what the bacteria or viruses want. For example, Agrobacterium tumefaciens, a pathogen of several dicot plant, is able to deliver a piece of DNA and non-ST DNA to transform normal plant cell into a tumor and direct these tumor cells to produce the